Yes, okay, great. So, we know that speciation is a process, and in the simplest sense, it starts with freely interbreeding populations and ends with two species that no longer exchange genes. But we also know that pre-mating isolation can evolve anywhere along this continuum, and that signals that are important in species recognition can continue to diverge long after the process of speciation is complete. So if we want to understand something about the evolution of pre-mating isolation, we do better to study it while it's incomplete or recently completed. So what I'm going to tell you about today is the first results of a study whose long-term goal is to understand the genetic basis of pre-mating isolation between two closely related species of mice. So what do we know about pre-mating isolation? Well, we know a lot, especially from taxa that use acoustic and visual signals. And so from these, we know that signals that are critical to mate choice and attraction within species can also play a key role in pre-mating isolation. We know considerably less about the genetic basis of pre-mating isolation. And notably, most of what we do know comes from insects, and particularly systems that use chemical communication. And so this is where mice come in. And so in mice, chemical communication really dominates. And we know quite a lot about the signaling component, thanks in part to the work of Jane Hurst and her colleagues. So we know, for example, that male mouse urine is this complex chemical blend that has these large proteins in it called major urinary proteins. And we know that females use components of male urine to get information, and that information can inform their mating decisions. And we also know that females just like male urine. They find it rewarding. They like to be around it. And so that's the signaling end. So how do they perceive this chemical bouquet? So that's where the vomeronasal organ comes in, or VNO. And so the VNO, if you don't know, it's this specialized pocket of olfactory epithelium that sits right above the soft palate. So we have one too, but it's completely useless. Not so in mice. In fact, in mice, it's essential to normal reproductive behavior and also gender discrimination. There have been upwards of 400 receptors identified so far in the vomeronasal organ, VRs, or vomeronasal receptors. And critically, these receptors are expressed in neurons that are a direct line of communication to parts of the brain that are critical in decisions related to social and reproductive behavior. And we know that these receptors are fairly finely tuned to different types of chemical ligands that are present not just in urine, but also other excretions. And we also know from the work of Catherine Dulac and others that individual receptors not only respond to specific chemicals, but they also respond to specific classes of individuals. So for example, males versus females, and critically for this talk, some subset of receptors seems to respond to heterospecific congeners, so to closely related species. And so collectively, this information about the VNO has suggested for some time that it might be a system that could be important in species discrimination and pre-mating isolation. And so all of this work that's been done on the VNO has been done in mice, in lab mice, which are house mice. And so it turns out that house mice and their closely related non-commensal species are actually a great system that has biological relevance to test the notion that the VNO might contribute to pre-mating isolation. And so you've already heard a little bit about house mice and their classic mammalian model for speciation. But there's 3 subspecies, and they're variously co-distributed with other closely related non-commensal species. So the pair I'm going to focus on today is one of the house mouse subspecies, Mus musculus domesticus, I'll just call them domesticus from now on, and Mus spreadus. And so the two are sympatric throughout the range of Mus spreadus in Spain and southern France and North Africa. And going back to our continuum of speciation, they are recently speciated. So they are what we think of as good species. But there is evidence for historic gene flow, however, none for ongoing hybridization. F1 males are completely sterile in both directions of the cross. So if females from either species were to mate with males from the other species in nature, there'd be a high fitness cost. But we know this about F1 males because lab crosses are still possible. And so it's actually quite easy to get a domesticus female to mate with a spreadus male and reproduce. The reciprocal cross is considerably more challenging, and there are a number of reasons for this, but one of these is potentially that there's an asymmetry in the strength of pre-mating isolation, such that spreadus females are potentially more discriminating than are domesticus females. And so, as I said at the beginning, the large question, my long-term goal is to understand the genetic basis of pre-mating isolation between these two species. And to take a first step in that direction, the experiment I'll tell you about today was a really pretty simple experiment. So we did this in the lab with wild-derived strains of mice. We took female spreadus and female domesticus, and we exposed groups of each to chemical cues from either males of their own species or males of the other species. So spreadus were exposed to either spreadus male stimuli or domesticus male stimuli, and the same was true for domesticus females. And the stimuli were this freshly concocted brew of urine and freshly soiled male bedding that we let the females interact with. And then we extracted their VNOs, sequenced transcriptomes, and we did this on the Illumina platform. We aligned the genomes to the transcriptomes to their respective genomes and tested for differential gene expression. So I'll cut right to the chase and give you the results for domesticus females first. First I'll tell you the questions that we answered. Let's get that done first. So the main question that I wanted to address is simply whether VNO genes are differentially expressed between females that are exposed to either conspecific or heterospecific male cues. And if so, what are the functions of these differentially expressed genes? Critically, are vomeronasal receptors differentially expressed? Because these are really the prime candidates for potential contribution to pre-mating isolation. And is there evidence for positive discrimination? In other words, are some genes specifically upregulated in response to heterospecific male stimuli? And because we were doing this as a reciprocal experiment, we also were interested in the difference between females of the two species. So we're exposing them to the same chemical stimuli from males of the same species, but do they respond similarly or differently when the male is conspecific from the perspective of one female and heterospecific from the perspective of the other? So now I will give you the results for domesticus first. So in answer to my first question, which is sort of the preemptive question, is there a difference between these two treatment groups? Not really, no. So this is a volcano plot, but it looks like a long dead volcano that has not, if it ever erupted significant data, it's done. And so what you're looking at is p-value corrected for multiple tests against the difference in expression for these two groups. So going in this way, there would be higher expression in response to the spreadus male stimulus in this direction, higher expression in response to the domesticus male stimulus. So we were able to measure and test for differential expression in 28,000 transcripts, and of these two were significantly differently expressed, and both were higher in response to the domesticus stimulus. So I'll just mention what these are since they certainly stand out there. So they're quite interesting. So ETD5 is a transcription factor that's previously been identified in a lot of male fertility phenotypes and also potentially in male reproductive behavior, but never in female behavior. And the second one is an immunoglobulin, which is also interesting because it suggests that there's potentially a very fast upregulation of immune response in females, potentially in anticipation of mating. We were able to identify 241 of the vomeronasal receptors so far, and none of these were significantly differently expressed. So this is, a solidly negative result that would have been very disheartening except it's actually quite interesting in light of what we found for spreadus females, which was quite different. So I'm showing you the same type of plot and the same direction of the comparison. So again, this is higher expression in response to spreadus, and this is higher expression in response to domesticus. But remember that from the perspective of the females, this is now reversed. So these are the heterospecific males, and these are the conspecific males. So we found 152 genes that have significantly different expression between these two treatments. And notably, 100 of these, so twice as many, have higher expression in response to the domesticus stimulus. So I'll just highlight what a couple of these are. So the first one is a steroid hormone receptor, and we're still working on annotations for a bunch of these genes, but the next one that we have an annotation for is indeed a vomeronasal receptor. The two that have the highest fold difference between the two treatment groups are also interesting. They're proteins that were identified in salivary glands in mice, so they're more associated with excretion signaling rather than perception. So there's obviously a lot more to look at here, but just to highlight on the spreadus side of things. So if the genes that are upregulated in response to the spreadus male stimulus, of the ones that we're able to annotate so far, all the top three with their most highly significantly different are all vomeronasal receptors. And so even though we've currently been able to identify with confidence fewer vomeronasal receptors in the spreadus transcriptome than in the domesticus transcriptome, we've already found that 8 of these have significantly different expression between the treatment groups for spreadus females. And so I'll just briefly show you these genes. So these are just bar graphs. This is mean FPKM, which is a normalized measure of expression for transcriptome data. And so the blue bars are expression when females are exposed to a spreadus male, conspecific male, and red bars are domesticus, heterospecific male. And so these 5 are all upregulated in response to a conspecific male stimulus. And this is cool, but this is exactly what we think that vomeronasal receptors are doing in general. We think that they're important in conspecific interactions. So what's more exciting for me is that so far we've found 3 that are significantly upregulated in response to the domesticus stimulus. And so these, although it's early days to say this, but they're potential candidates for involvement in species discrimination and contribution to pre-mating isolation. So to sum up what I've told you in these very fetching species of mice, for domesticus, we found a minimal difference in VNO response in these females exposed to conspecific or heterospecific male cues, no differential expression of VRs, and no evidence for positive discrimination of heterospecific males, so no upregulation in the spreadus treatment. In contrast, in spreadus, we found this large difference in VNO response in females exposed to these different cues. And we found a subset of vomeronasal receptors that are among the genes with the largest differences between treatment. And strikingly, there seems to be evidence for positive discrimination of heterospecific males. So we have twice as many genes that actually have higher expression in the heterospecific treatment. And I'll just close by saying that this difference between the two species, between the female's response, is actually potentially quite interesting. And so I'm still trying to figure this out, but one biologically interesting explanation is that it may reflect, as I kind of suggested before, a difference, an asymmetry in the strength of pre-mating isolation between the two species, such that spreadus females are more discriminating than domesticus females. And so with that, I can't believe I got through this time. This is a first. I thank my lab group, my funding sources, people that were generous in sending me mice. I really need graduate students.